All right, so we just finished covering the trichromatic theory of color vision. Now we're going to talk about another theoretical approach to color vision that is called the opponent process theory. This theory was first proposed by a guy named Herring in the 1800s. And basically, what he did was instead of having people look at different color lights and measure what they were perceiving through those mixing of those different colors. What this guy did, what Herring did, was he had people do phenomenological descriptions of what they were perceiving, and through these behavioral descriptions of what people were perceiving, he came up with this opponent process theory of color vision. And essentially, what it is that color vision is controlled. Color vision is caused by different processes that oppose each other, that work in opposition to each other. So, for example, there's one process that mediates the perception of the colors blue and yellow. Whenever you view something that's blue, this process produces less output. Whenever you view something that's yellow, the process produces much higher output. If a receptor perceives something that's got blue and yellow in it, then the output's somewhere in the middle. So you have this one process that reduces its output when it sees blue and increases its output when it sees yellow, and is in the middle somewhere with blue and yellow. Similarly, there's another process that is mediated by green and red. So whenever it sees red, whenever a receptor attached to this process sees red, it increases its output. And whenever it perceives green, it reduces its output. And if there's some mix of green and red, then there's a middle level of output. So you have these opposing processes working against each other, but they're connected. The blue and yellow process are connected, and the red and green process are connected. And that, in essence, is the opponent process theory. Some of the behavioral evidence for the opponent process theory. Well, first, color blindness. Most types of color blindness are either red, green, or blue, yellow. We talked about some of those types of color blindness earlier in this lecture, and you notice that people either had trouble perceiving red and green or blue and yellow. The other evidence of for the opponent process theory, it comes from looking at images like this. Now, this image is in your book. Basically, if you stare at the cross in this image for a very long time, I think for like a minute or something like that, and then look away to a white blank area, you should see an after image of the proper colors of the American flag, red, white, and blue. What's happening is, as you stare at that cross, you're burning in the colors green, black, and yellow into your retina. So those processes, remember, the process that mediates the color green is also attached to the red color, to perceiving red. The process that mediates the perception of yellow is also attached to blue, and the process that mediates the perception of black is attached to white. So what you're doing by staring at this flag is you're driving those three receptor types to one extreme of the process. As you stare at that cross for a minute, you're burning that into your retina. As soon as you look away, the receptor, the opponent process bounces back the other way because it's no longer being pulled by the yellow, by the green, or by the black. It gets thrown the other way. So for a brief moment in time, for 1 or 2 seconds, you actually see the opponent process color. So where you're seeing yellow, you see blue. Where you're seeing black, you see white. And where you're seeing green, you see red. Okay, so here's another example. This is something you can do at home, right, with the picture that's in your book. So this image should be in your book. What you do, You start with the green square, which is right there, right? So what you do is you cut out a little half-inch piece of paper and you put it right there in the middle of the green square, and then you cover up the other squares, and then you stare at the green square for a minute or so, and then look away, and you should see an after image. Do the same and write down what colors you see, and then do the same with the other squares. So then cover all these squares and then put the little white square in the middle, and then stare at it and then look away. Write down what you see. Do it with the blue and the yellow. What you should see is something like this. So in the green square, what the outside after image, so the part that was green outside the little white box that you put there, should be red. So the red flips over. Inside the after image, you should see green because of the fact that that white area is being surrounded and stimulated by green. You should see green on the inside. Similarly, with the red, you should see the opponent color of red, which is green, and then you should see the color red. And the same goes with the blue and the yellow. So here's what Herring proposed, right? He said that there's three opposing mechanisms working. Red-green opposing mechanisms, blue-yellow opposing mechanisms, and white-black opposing mechanisms. The pairs respond in an opposing fashion. Like, for example, the red-green one fires, or whenever it perceives red, it fires a lot more. And then when it perceives green, it is inhibited. It does not fire. And of course, what Herring, this was in the 1800s, so they didn't know about, the structure of the retina back then. But they were smart enough to know that the retina had something to do with vision. So the proposal back then was that the opponent process was due to some chemical reactions that were in the retina. And here's a cartoon drawing that shows a particular process and the opponent colors that are associated with each one. So this is the black and white one, right? So whenever it perceives black, it is inhibited. Whenever it perceives white, it is activated, and it produces more output. Similarly, this one is excited when it sees red. It produces more output, and inhibited when it sees green. This one is inhibited when it sees blue, and is activated or excited when it sees yellow. And they work together. They work in opposition to each other, right? So if this one's being stimulated with more yellow, it's going to necessarily inhibit the blue. If it's a combination of blue and yellow somehow that's being stimulated here, then it'll be somewhere in the middle. There is actually quite a bit of physiological evidence for the theory of opponent process. Back in the 1950s, we were able to identify opponent neurons. In other words, neurons that seemed to behave like this. We actually did record neurons that would fire a lot, single neurons that would fire a lot when they're stimulated by red and not fire at all when they're stimulated by green. These retinons were located in the retina and also in the lateral geniculate nucleus, which is one of the first relay points of visual information. One of the first relay points that visual information hits when it goes into your brain. And basically, like the cartoon processes that are illustrated here, these neurons behave the same way. So here, here's an example, a cartoon drawing of some of the data. The columns represent different types of opponent neurons. So this is a neuron that is stimulated by blue and inhibited by yellow. And this right here, all of these little charts here, represent a neuron that's stimulated by red and inhibited by green. And if you just kind of record them when they're just sitting there not doing anything, you get this baseline activity. If you fire blue at them, 450 nanometers blue, of course the one that gets stimulated by blue goes crazy. Lots more spikes. and there are even less spikes seen in the red-green neuron. The green, if you give shine green light, 510 nanometers on these neurons, well, it doesn't have too much of an effect on this guy. But look how it reduces the firing of this guy. It goes from this baseline activity here to just a couple of spikes. Yellow, 580 nanometers, causes this neuron to fire less because it's inhibited by yellow. So it goes from this to this. And it also affects this as well. Not as much, but it does affect that one. And then finally, red. Again, it affects this one to a certain extent, but look how much it affects this one. A lot, right? The firing goes from this to this. So we have actually been able to identify neurons in the central nervous system that do behave a lot like you would expect opponent process neurons to behave. So what happens when we try to combine these theories? Well, they each describe different mechanisms in the visual system, right? The trichromatic theory does a really good job of describing the responses of the individual cones in the retina. So we're pretty sure that individual cones contain the proteins in them that seem to be sensitive to specific wavelengths of light, to specific regions of light, red, green, yellow, whatever. On the other hand, the opponent process theory explains how some of the processing happens after the cone. So the cones feed other neurons in this chain of neurons that ultimately leads into your brain. And so one of the thoughts is that the opponent process theory may help us to understand how the logic works in the processing of information that comes out of the cones. So here you go, right? Here's a cartoon drawing of what I just said, right? So the trichromatic theory does a really good job of explaining to us how the receptors work. And in fact, it probably has something to do with color matching. Whenever a receptor for red gets stimulated, that gets activated. But the receptors probably feed their output to they do feed their output to other neurons, and these other neurons in the retina and then in the brain, or in the retina, I should say, the ganglion cells and the bipolar cells that the receptors connect to. These form circuits in the retina that probably mediate this opponent process, that probably control this opponent process, and ultimately feed their output to the brain. So here's a cartoon representation of that, right? This would be the wiring of some neurons that are sitting inside of the retina. In this example here, we would see a cone that responds to medium wavelengths and a cone that responds to long wavelengths. So a cone that responds to green and a cone that responds to red. And it turns out that those cones probably may feed an opponent process cell that looks for green input, and whenever it gets green input, it shuts down. And it also looks for red input, and whenever it gets red input, it increases its firing. We also have these types of another circuit for the blue and the yellow response. So once again, we have a cone that responds to the shorter blue wavelengths, and we have the cone that responds to the medium green wavelengths, and a cone that responds to the longer red wavelengths. This circuit's a little bit more complicated. Whenever green or red is perceived, it stimulates this neuron here, but then this neuron goes and it inhibits this neuron. The end result being whenever something in between the medium and long wavelengths is perceived, that is yellow. Whenever that's perceived, it inhibits the firing of this opponent process cell. Or whenever blue is perceived, it causes this particular cell to, or it stimulates this particular cell here, and it causes it to increase. So what this circuit ends up doing is causing an increase in firing whenever blue is perceived, or it causes a decrease in firing whenever yellow is perceived. Okay, so one question you may have is, how do these receptor neurons and opponent process neurons work together to determine the differences between different wavelengths? Well, this picture here kind of gives you that idea. Here we have the two exact same circuits in A and B. Notice we have a green receptor and a red receptor, green receptor, red receptor, both of which connect to this other neuron, this opponent process neuron. Now notice that in both of these circuits they're identical. So in this circuit the green one always inhibits this neuron, so it causes it to shut down, and the red one always increases the firing of this neuron, so it excites it and causes it to fire more. So you've got this same circuit, but you can use this same circuit to detect two different wavelengths of light. In this example here, we have 500 nanometers going to this circuit and 600 nanometers going to this circuit. As you know by now, 500 nanometers is closer to green. So what's happening here, whenever 500 nanometers, which is greenish, hits this circuit, it fires up the green photoreceptor a lot. And a little bit of the red as well, right? But mostly the green. So the green is driven to minus 80 in this example, whereas the red is driven to plus 50. And when you sum those outputs together, you get an output of minus 30. So what's happening here is that this circuit is working together to reduce the firing whenever it perceives green. This same circuit can also work together to increase firing when it perceives red. And the way that works is you shine a light of 600 nanometers onto these receptors, so that causes this one to reduce, since this was an inhibitory connection, it only inhibits down to 25. But this is an excitatory connection. It's going to stimulate this neuron up to 75. And when you add those two stimuli together, you end up with a plus 50, which tells us that by looking at the output of this neuron, we can easily tell what is being perceived by this circuit. So whenever the output is minus 30, we know we're looking at something we know it's perceiving something that's green. Whenever this output is looking at something that is red, we know that the output is going to be plus 50. And remember, this is opponent process, so red and green work together, right? But this is one way in which we think the opponent process cells may work together to allow us to detect different colors with the same circuit, the opponent process circuit.